This podcast is brought to you by the School of Advanced Study, University of London.
Good morning. Let me welcome you to the Institute of Philosophy and the Centre for the Study of the Senses. We're delighted to have you all here, and especially to have such an impressive list of researchers working on olfaction and related issues. We at the Centre have been trying to pioneer relations between philosophy, psychology and neuroscience, and we have succeeded to some extent in at least convincing the AHRC to provide a substantial grant to bring this work forward. And here we've got many of our partners and collaborators attending today and attending many of the other workshops. So I hope that all of you will keep in touch after today and that we can continue to try to press forward some of the foundational issues. There's an awful lot of results that are coming out of contemporary neuroscience, huge proliferation of results, but at the same time it's a little hard to see what the underlying theory is. We're often in a situation a little bit like Darwin before the genes, and it would be very good to figure out what some of the underpinning models or foundational planks would be, and I think this is something which those who are empirically informed philosophers and those who are philosophically inclined neuroscientists can do a lot with by working together. So to start off today, let me just say, if you haven't signed in, please do sign in. We'd like to keep a record of who's here, and we're going to be filming the day, so if you haven't signed consent forms, please do that, because otherwise we can't put anything online because of the Data Protection Act. Now, the first session this morning, I'm very glad to say, is a session with J. Gottfried, who we have been hoping to bring to London, and especially to the centre, for a long time. There's quite an interest in taste and smell here, work on flavour perception, but we've become increasingly interested in smell itself and on the issues surrounding it. And Jay's work has obviously been one of the sort of leading pieces of research that have driven us into the questions that we've asked here and the ways in which we've tried to pursue issues about how smell works, how important it is, how it connects to other aspects of mental life, including the emotions, including memory and cognition. And when we look at this work, I'm glad to say that we can locate Jay in a certain space. One of his advisors was Anjan Chatterjee, who is a neurologist who's turning himself into something of a philosopher with his pioneering work on neuroaesthetics. And then another great collaborator with Jay is Ray Dolan across at the Functional Imaging Lab, who's also been a strong supporter of the project. So it's about time we welcomed you into the family. We're glad to do it. So Jay, can you start us off with function follows form, the shape of smells, the admiring olfactory system, and coli Sidebar. Thank you. Was it on before? It's coming. If I put the lights down, you'll be able to see you better.
Thank you for that very gracious introduction. It is very exciting to be here. I love coming back to London. And I like what you said, Barry, about the philosophically minded neuroscientist. I would put myself in that category. And as you'll see, I'm going to try to provide a broad pastiche of what the sense of smell really means from various different kinds of perspectives. There will be plenty of neuroscience for all of you geeks out there. But this is a process, or as I learned, process. That's how you say it here. Anyhow, E. coli, Sidebar, the whole works. So, and I really cherish this idea of unifying, trying to unify philosophy and neuroscience. I think that there's such a balkanization these days in science in general, where everyone has their own very specific niche. People rarely are stepping back and trying to consider things in a more holistic and comprehensive perspective. So this is one of the goals I have here. So this is my outline. It's a graphical outline. I'll speak a little bit about the philosophy of olfaction briefly, because I don't want to embarrass myself too much in front of this crowd. A few words about the neurological, something about the evolutionary, which really relates to what is the function of an olfactory system. And then from there, discuss what I call the sensory ecological. So what is the form of the stimuli that are important in the environment that the nervous system needs to be able to process? So it's really it's a well choreographed development between the stimulus and the system. And then finally we'll get to the neuroscience. And I'm sure you'll all agree that perhaps the most unifying principle will be something from the pages of Wonder Woman comic books. So I couldn't resist. Maybe it's my jet lag disinhibition, but this is such a beautiful crystallization of why smell can be so useful. So Wonder Woman gives Agent X a clever imitation of the real papers. I'll give you 15 minutes to bring me Diana White as agreed. If she isn't here, I will hurry. Wait here, Wonder Woman. So she's sitting in this room all by herself, bound to the chair. Wonder Woman's super keen sense of smell detects a faint odor of anesthetic gas in the room. They're trying to gas me. I'll have to hold my breath until they think I'm knocked out. So here, Wonder Woman is using this, her sense of smell to alert her to imminent danger. And one interesting point that is worth bearing in mind is that she's going to hold her breath. She holds her breath while she's not going to be absorbing the gas, but she's also not going to be smelling the gas. And this is just to illustrate that in order to smell things, you need to participate in that process by making a sniff. Okay. There's a very ancient philosophical tradition of exploring the senses. One guy who tackled this was Aristotle. He got some things right, some things wrong. He was a product of his time, and his time was all about humans as microcosms of the universe. So we have the four elements: fire, air, water, and earth. And he tried to relate the different senses to these elements. You may notice that there's only four elements, but we have five senses. He had trouble there. Does anyone want to suggest which of these senses was olfaction, vision, auditory, and so on? You could do this guessing game if you want. Avery,
was air smell? Yeah.
No. No. So smell is fire, right? Like think about all those ancient traditions where they're doing burnt sacrifices, sending all those aromatic signals up to God. So smell was fire. Hearing was air. This is kind of a morbid one. Vision was water, because they find that the eyeball, as it started to rot away, would release its vitreous fluids. So it seemed to be a very watery kind of sense. And then touch was earth. And then Aristotle decided, well, let's just throw taste in there too, because there's a tactile component at the time, and so on. He did write specifically about smell, and he had some interesting ideas which will actually come back a little bit later in some of the recent work we've been doing. And he had this idea of conditional pleasant and unpleasant smells. So there's a context. He said their pleasantness and unpleasantness belong to them contingently, for since they are qualities of that which forms our food, these smells are pleasant when we are hungry, but when we are sated and not required to eat, they are not pleasant. Neither are they pleasant to those who dislike the food of which they are the odor. So this was a kind of advanced notion at the time, and although it's a little bit archaic in the Latinization of this description, I think it's still very pertinent to basic models of olfaction that we're using today. So neurologically, I could tell you about how Alzheimer's disease and Parkinson's disease are among the many different neurodegenerative diseases where one can lose their sense of smell very early in the process, sometimes even before the emergence of other overt signs and symptoms. And so there's a fairly active effort these days to try and explore whether we can use olfactory measurements, either behavioral or imaging, to try and develop pre-diagnostic biomarkers to trace these disorders. But I'm not going to tell you about that. I'm going to show you these men from 1907. And it's a question for the audience. At the time of this photo, what distinctive odor was likely to be detected on the breath of these patients and why? So as a neurologist, one would want to assess the patient, maybe apprise yourself of what their probable profession is or was. Can say they're all in an asylum now, unfortunately. Looks like they had purchased nice clothes but haven't updated those in many years. They look like the men about town. They have no wedding bands. Any guess as to what the diagnosis might be? They're mad.
To use the technical term.
Right. Bachelors.
Homosexual.
Well, they are demented, which, yeah. So these guys had dementia paralytica or neurosyphilis. And the treatment back then was arsenic, which probably worked. It just killed you, but it probably worked. And that left a very characteristic odor on the breath, which is garlic. One other quick one here. These patients were asked to look at the camera. Which of the following olfactory perceptual features do they have difficulty identifying? Oh, sorry. Yeah. So these patients also in the asylum are very contorted and twisted up. They have trouble controlling their movements, regulating their movements. They have ballistic movements. These patients have Huntington's disease, and they characteristically have trouble detecting disgust. The last one here, because we are in London, this eminent 19th century English neurologist was among the first to describe olfactory auras in epileptic patients. Who was he? Jackson, Ferrier, Gowers, or Voldemort? So John Hewlings Jackson, very famous neurologist who really established a lot about epilepsy and epileptic olfactory auras, and he took many different drugs. Okay, the evolutionary side of things. The function of sensory systems, in my humble view, is the following: to extract behaviorally meaningful information from sensory stimuli in the environment. Obviously, we're detecting all sorts of things in the environment that may not be meaningful, but the goal is to be able to extract important information from the environment. And so, by translation, the function of chemosensory systems would be to extract behaviorally meaningful information from chemical stimuli in the environment. Now, if you consider this the great blue sea of behavior, everything from maternal bonding, hunting, feeding, territorial marking, escaping predators, kinship identification, mating choice, for many, many species have at their root odor, the great unifier, right? So olfaction is really driving much of fundamental behavior in the great majority of animal species. This is true even for an amoeba in some mountain lake. Here it is kind of drifting around, using chemical gradients in the water to guide itself towards nutritive chemical compounds or to escape noxious compounds its own waste, for example, using the chemical senses to obtain what it needs. Here's our E. coli. What's fascinating here is this is a unicellular organism, obviously, and what you can see at the arrow is radiographic staining of a protein complex. This is a chemoreceptor complex. Essentially, this is a nose for the bacteria. It's localized to one end. It gives the organism polarity, so it has a way of navigating appropriately through the space that it lives to hone in on foodstuffs. You can even kind of imagine in E. coli the sense that there's already a fundamental dichotomy between the pleasant and the unpleasant. So this is really great bacteriology work in the 60s and 70s by Adler and colleagues. On the left side, what he's done is put in a nutritive amino acid into this pipette tip. And you can see the bacteria swarming to the tip of this pipette, kind of engendering a very fundamental arousal approach response. In turn, on the right side, he's taken an organic aliphatic acid and plated it from about 1 o'clock around 12 in increasing concentration. And you can see that the bacteria are clearing away from this compound with increasing concentration, a kind of very innate avoidance response. So here we already have the kind of pleasant, unpleasant dichotomy that we all naturally experience with smells. And of course, smell has great relevance for human behavior. It's, of course, very important for food and for threat, and also sex. Sometimes that gets a laugh. All right, let's move to the sensory ecological. So again, we've sort of established some of the basics of an olfactory system, what it needs to be doing. We don't need to smell our way through traffic, but we may need to use the sense of smell for guiding us towards or away from behaviorally meaningful stimuli. So there's a concept I like to, that helps me organize in a pseudo-philosophical way how I think about these systems. And the idea is that the stimulus shapes the system. So one way of posing questions, what sensory ecological forces have shaped the evolution of olfactory systems? Or what is the natural form of odors typically encountered in the environment? There's a really amazing book that came out over 20 years now by David Dusenberry about sensory ecology and what is the physical, chemical, structural forms of sensory stimuli that sensory systems need to be able to grapple with. Here's a neat example. In order to achieve, so let's say evolution got a bit stuck and the original visual plan of the compound eye in insects remained kind of the primary visual detector apparatus. This is based on kind of optics and physics modeling showing that if we wanted to achieve the kind of spatial resolution and depth perception that we all enjoy and love, if we were endowed with a compound eye, that's the size it would have to be. So you can see that obviously with our lens-based eye, nature came up with a different kind of solution to the problem, much more efficient. So the form of odors. What form do they take? What do natural odors look like? Well, first of all, it's important to bear in mind that there's really a limitless variety of molecules that could contain, that could carry smell. If you just consider one molecule here, this is a 7-carbon chain molecule, heptane. It has a kind of ethereal, fruity-ish odor. you can see that you can modify this molecule in dozens of ways, and each one of those may take on a slightly different to very different kind of olfactory characteristic. So the olfactory system needs to be very flexible and adaptable to be able to detect, encode, and interpret a huge number of different possible combinations of smells. The other important thing to bear in mind is that most odors aren't single molecules. So here's a cup of coffee. How many different kinds of molecules, odorous aromatic molecules, make up the odor of coffee? Any guesses?
631.
Okay, well, except for berry and the other guesses. Yeah, so 500 range. Yeah, so, but what's fascinating is we don't detect the elements, those 600 and something elements. We detect the whole, right? Coffee. And this gives us, essentially what we're doing, we're encoding information about that whole object that we're confronted with. And it gives us a lot of flexibility that way to generalize across different coffees and say, oh yes, we can form this category, they all belong to coffee. Yet we still do retain discriminative capacity to distinguish maybe the Kenyan blend from the Java blend and so on. And we see this sort of thing playing out even in ants. So here's a poor carpenter ant, which has like been glued to a piece of cardboard, and they're presenting the smell of a close friend of his that he's grown up with in the ant colony. And he's very chilled out. But now you give him a non-nestmate extract from the same species, and now it's, claws out, fangs bared. He's very unhappy here. And they use a technique called gas chromatography and mass spectrometry to characterize the components of these nestmate extracts. And you can see that there's 25 or more compounds which together generate this kind of, approach or avoidance based response in these ants. And if only this poor lady had that formula at hand when she was being attacked by that ant. Okay, let's move to the neuroscience. I think it's important to say something about anatomy. I found that most of my colleagues, even after I give several talks that they've heard, fail to remember the olfactory anatomy. But I'm sure you won't be one of those. So this is a cartoon schematic, sagittal section. And what's important to note here is when you smell an odor, it enters into the nasal cavity, and these are called turbinates. They're little cartilaginous shells. It helps create a lot of turbulence as you inhale the odor through the nose, and helps warm the odor, which enhances binding of the smell to its receptors, which are sitting in the very upper reaches of the nose. Short of doing major surgery, there's no way you could actually touch those receptors with your finger. So here we have a bunch of odors circulating through these turbinates, and they bind to the endings that you can kind of see poking through. The red slab, the horizontal slab, is the olfactory bulb, and the bulb connects to the olfactory receptor neurons, which send their axons through what's called the piriform plate, and synapse there in the bulb. This is kind of a close-up view of what's happening at the epithelium in the back of the nose. Here you have your olfactory sensory neurons. These are supporting cells in green. The sensory neurons have a kind of dendritic knob, which then sends out cilia to spread in a mucus layer at the surface of the epithelium. And this is where the olfactory transduction takes place, where the odorants bind to receptors expressed on these cilia. And then from there, the message passes to the olfactory bulb. And these are just simply amazing freeze fracture electron microscopy images. This is the epithelium. Look at this kind of dense mossy carpet. Those are all the cilia. These are the density of olfactory receptors there is just astounding. Here's an olfactory receptor neuron, right? And you can see there's that dendritic knob coming up. There's another one, there's a knob, all those cilia, just a dense mat. This is 4,000 magnification. And then look at this at almost 13,000 magnification. This is at the end of one stop, those cilia branching out. Just spectacular. It's sort of like nature recapitulates itself. These look like sea anemones in the ocean. So really cool stuff. So if we zoom in on this bulb area and flip things upside down, this is a human brain, which is my particular animal model we study. You can see the two olfactory bulbs. They look like earthworms run over after a rainstorm on the road. They're very flat. So we have our olfactory receptor neurons projecting information to the olfactory bulb. This then disseminates information to a variety of structures in the basal frontal. So just to orient you, ventral view of the brain, right? So looking from below, the eyes would be towards the top, back of the head down here. Here's one temporal lobe. The other temporal lobe we've cut in cross-section to expose kind of these olfactory structures, which are very deep and limbic. So the bulb projects to a variety of structures, including the piriform cortex, which is encoding odor object identity, so mintiness, strawberryness, wet sidewalk after rain smell, stuff like that. The bulb projects to the amygdala, which is likely extracting emotional valence features from an odor. The bulb projects to the entorhinal cortex, which is just one synapse removed from the hippocampus. So this is thought to be involved in kind of the mnemonic interplay between the stimulus and encoding consolidation mechanisms. All of these regions project anteriorly into Barry's favorite structure, the orbitofrontal cortex, where a lot of the integration seems to occur. And I'm not going to talk about taste, but this is the place where all the different elements of the perceptual elements of the odor appear to be bound together to form a more rich, consolidated percept. And then there's also a lot of feedback connections to help kind of reshape and modulate the incoming input. And three important points to bear in mind about the olfactory system is, first of all, there's tremendous overlap with a limbic affective system. And as I said before, based on the idea that olfaction seems to be so critical to many animals in terms of behavior. In some ways, what the human brain has been able to do is kind of co-opted an olfactory structure to engage with emotional processing and behavioral control across all of the senses, which is what it does. One curious fact about the olfactory system is there's no thalamus involved. So for all of you sensory experts out there, you've probably learned that essentially all of the senses have to pass through the thalamus before that information reaches the cortex. And this variously provides ways of filtering the signal or parsing the signal or bundling into different kinds of perceptual streams for later processing. But in the olfactory system, you don't need to go through the thalamus in order to reach cortex. Whether this means that the olfactory system simply doesn't need a thalamus, maybe it evolved before the whole kind of thalamocortical module arose in the brain, or maybe another region of the olfactory system like the bulb could perform the effective functions of the thalamus. There is a conduit to the thalamus. It's an indirect path from piriform cortex and the amygdala to the medial dorsal thalamus, and then to the OFC, or low frontal cortex. But it's not very clear exactly what role that plays. And the last thing, as I mentioned before, that a lot of these areas are curiously susceptible to neurodegenerative diseases very early in the process. No one really knows why this is. There are theories that maybe a lot of these diseases are ultimately triggered by toxins or other compounds that enter through the nose. This is a close-up here. It's a little bit, perhaps, early in the day for this, but again, ventral view. Here's the midline. The eyes would be up there. So the olfactory bulb converges to the olfactory tract, swinging down and around. There's our orbitofrontal cortex. And this is quite striking. You can see these different fascicles from the bulb. Looks to me like they're going to different areas, but it's very difficult to trace areas in the human brain. But you can see that this lateral tract seems to be headed right towards the piriform cortex. These medial tracts likely are headed into basal forebrain areas. And what I love about this picture is it shows a region that in most anatomical textbooks you don't even really see. This is the basal forebrain, and these holes or perforations is known as the anterior perforated substance. And this is essentially the area where we have the nucleus basalis minor, a dense concentration of neurons containing acetylcholine, which then is broadcast through much of the brain. And these cholinergic cells here, they generate Alzheimer's disease. This is a very such a small structure, so buried there, but it's essentially important for attention and memory. And here you have a first-hand look at where it is. All right. The rest of my talk, I'm going to discuss a few recent studies of ours. I'd like to begin just by discussing how can we measure odor information in the human brain. For a long time, it was difficult. Any kind of cognitive neuroscience generally was based on lesion models. Right? You have a patient with a lesion, whether through the vagaries of nature or through a surgical approach like this one, where an epilepsy patient had pretty much the whole medial temporal lobe taken out. See that big black hole there? And so you could correlate various, whether it's visual or olfactory or anything. Marilyn Jones Gottfried and Robert Satori did a series of studies showing that there were resulting deficits in odor memory, odor identification, detection, and so on with lesions like this. But as I showed you before, because there's so many regions clustered into this area, it's really hard to make more definitive statements about what area might be involved in a given olfactory test. Now there's another approach using electrophysiology. Because a lot of these structures are deep, the olfactory structures are deep, using surface EEG recordings can be tricky because those sources are so deep, it can be difficult to definitively interpret where the source of those signals is coming from. But there are some opportunities to put electrodes directly into the brain. This was actually pioneered by Penfield in the 40s. So you can record directly from the surface or from within the body of the brain. But these are rare patients and not really capable of generating large-scale data sets. So really since around 1990 or so, we've had functional imaging, which really has revolutionized our understanding of the human olfactory system and more generally the human brain. And the idea is that we begin with neuronal activity, whether you're looking at a picture or smelling an odor or remembering your grandma. You have neural activity, and this is energy demanding. There's no energy needed at the moment firing off action potentials and broadcasting that message. But you need to regenerate the stores that you've just depleted. So there's the idea of massive energy expenditure, which needs to be reclaimed. Okay? So because the brain needs oxygen to create energy, it's not like a muscle where it can use a variety of different sources of energy. You need ATP, which means you need oxygen. And how do you get oxygen to the cells that need it? You carry it through the blood. And essentially what we're detecting using functional imaging are these changes in blood flow, blood volume, and oxygen levels. And these were on the basis of PET scanning and functional MRI. So these are surrogate markers of brain activity. Human olfactory functional imaging. This is back in the days. This is at the Phil, right down the street. 3 odor experiment, and the 4th is a blank. Air passes through these chambers, which have a saturated liquid pellet of odor, and then that's carried to the subject's nose, and we can switch back and forth between one valve and another to direct odor or move to odorless room air. Now, research in my lab, very broadly speaking, is interested in understanding how sensations at the nose are transformed into percepts. And a lot of our work focuses on understanding how learning, memory, experience, and context shape our perception of the olfactory world, such that maybe this hamburger takes on more of a characteristic of steak. And we're using many different techniques. We're using high-resolution functional MRI, olfactory psychophysics and behavioral approaches, physiological monitoring, multivariate pattern analysis. This is a newer method that allows us to consider not just where something's active in the brain, but a little bit more about how it's encoded. And we can use these techniques to consider spatially distributed ensemble patterns, which provides a new way to identify feature-specific signatures in the brain. We're also using computational models, and we're starting to do some of this intracranial EEG recordings. Very briefly, the initial studies on olfaction were done using PET, and these studies essentially established, kind of replicated what had been known for the lesion work. So we're seeing odor active areas in the medial temporal lobes, in the temporal cortex, and in the orbital frontal cortex. And a variety of different labs were able to demonstrate the feasibility of this. PET scans are limited. You can only scan a given patient so much because you're using radioactive material. And you're confined to using blocks of trials, stimulation blocks rather than kind of very brief periodic events. Some of the original fMRI studies from people like Noam Sobel showed that the olfactory system is responsive not only to odors but also to sniffs. The sniff kind of primes the system, and this has been shown in animal models. The sniff alone is able to activate a lot of the olfactory centers. And if you give a subject topical anesthetic in the nose so they can't really feel that airflow moving through the nose, you reduce that response. So there's something, it's fairly unclear what is the detector in the nose that allows this to take place. Is it the 5th nerve, trigeminal nerve, that's sensing the airflow? Are there specialized neurons in the olfactory sheath that may be sensitive to these sorts of kind of mechanical changes? One other notable feature is that there's a lot of habituation in the olfactory system, and if you give a prolonged stimulus, the system adapts, and you don't find any activity. And what Sobel and colleagues did is model a kind of decaying time course to show that, in fact, there is activity there, only it rapidly decays. Now, when I got to Ray Dolan's lab, to set up the olfactory work. At Phil, we did a series of different studies. We found that different subregions of the piriform cortex, this is the olfactory cortex, respond differently to smells. We implemented a series of different learning and memory tasks to show that not only some of the kind of canonical learning memory structures were involved in these processes, but also piriform cortex. This study, for example, we had subjects learning associations between pictures and different smells. Maybe smell rose and see a helmet, and subjects would create a very brief mini vignette for themselves. Poor soldier fell on a battlefield in a field of flowers. So they did all this, they learned a series of different associations, and then the memory test, we just presented them with the pictures. The idea being is we might be able to reactivate olfactory information. What we find is when we simply present the pictures, we get activity in the piriform cortex, suggesting that there may be some kind of cross-modal reactivation. Now, I mentioned this multivariate pattern analysis. This is one of the first studies we published in this area. Very simple here. We present subjects with 4 different smells, each of which have a unique olfactory note, mint, rose, banana, and lemon. And these are 2 different subjects, and you can see that where we present a flattened map of the piriform cortex, you can see that there's a lot of overlap of these 4 odors within piriform cortex, but each odor is evoking kind of unique pattern. If we just average that activity across the whole piriform, we actually might not find any differences. But by using these pattern techniques, we can now pull them apart. One interesting thing is from one subject to the next, there doesn't really seem to be any kind of consistency, which speaks partly to the fact that there's a lot of anatomical variation from one subject to the next, as there is in the visual system too, especially V1. But also that each person has their own unique set of experiences by which they've learned about the olfactory space. So it stands to follow that there may be different kinds of patterns for each and every one of us.
It looks as though subject 1's from Italy and subject 2's from Corsica.
Yeah, I think it's Sardinia, but yeah. We've examined categorical perception of odor objects. As I mentioned before, maybe different kinds of coffees, but we can put them all together. We did a study where we presented 9 odors subjects, 3 minty, 3 woody, and 3 citrus. And you can see this is a cluster analysis showing that behaviorally subjects clustered each of these sets of 3 into the appropriate categories. And then what we did is we collected 2 kinds of similarity ratings. One was behavioral. So for each of the 9, we gave them pairs of odors and said, smell these 2, try and focus on their quality, how similar they are. And you can see in this 9x9 matrix, for the within category comparisons, there's lots of blue, they found them very similar. But when they were smelling 2 odors that were from different categories, they were rated as less similar. So that's fine, we have this similarity matrix there for the behavior. And then we did the same thing with the posterior piriform cortex fMRI activity. So again, we had a pattern in piriform cortex for each of these 9 odors, and then we basically just did a correlation between each possible pair and set up a matrix here as well. And so in the final step, we used something called multidimensional scaling to collapse this multidimensional space into a more tractable 3D space and determine how well the behavioral map overlapped with the neural map. And what we find is that, first of all, within a given category, whether it's woody, citrus, or minty, that the behavioral and the neural maps cluster very closely together. And at the same time, for individual odors, the behavioral and the neural map were fairly close as well. suggesting that this region of the brain is very much involved in being able to represent categorical information about a smell. And we didn't see this in other areas of piriform or amygdala or OFC. So as I said before, we've done a lot of work with odor discrimination learning and find that both posterior piriform and OFC are involved. Here's a study that in which we basically presented subjects with an odor for 3.5 minutes, a kind of passive exposure to provide them with the kind of sensory experience of a particular flower smell, let's say. And what we found is from before to after exposure, subjects are now better able to tell apart different flower smells, and we find that regions of the orbitofrontal cortex are involved in mediating this process. This work was done by Gwen Lee, a postdoc who's now at University of Wisconsin-Madison. She did a second study where we took 2 odors that smelled virtually identical and then paired one of the 2 of those with an electric shock. This kind of aversive conditioning resulted in the subjects now better able to discriminate between those 2 odors. And at the same time, we found in piriform cortex a fundamental reorganization of the pattern such that the once indistinguishable odors were now being coded more differently than before. So with these sorts of studies, we decided it would be fun to test the inverse question. If odor experience is important to enhance perception, what if we rob subjects of that experience? So what is the role of afferent sensory input in sustaining perceptual codes in the brain? And here's one of my more sophisticated movies. So I've shown you that, let's say, a rose will induce a rose representation in the brain. And the idea is, let's say we plug the nose. We'll plug it for a week, thereby robbing them of the sensory input. Maybe that percept fades away or becomes less salient than before. So Joanna Wu, who didn't realize how ambitious an undertaking this would be, or she wouldn't have done it. This was a 2-week study. So we admitted patients to the research wing of the hospital so we can monitor them while they had their nose plugged, actually taped over. They could sleep at night without the tape because you can induce toxic shock syndrome if you do it for too long. Anyhow, baseline behavioral testing, functional scanning, and endoscopy procedures to take a look at the nose to make sure there wasn't any edema or inflammation arising. And then they stayed in the hospital for 7 days. It was kind of an odor-free environment. After 7 days, we repeated the testing and then brought them back 7 days later after recovery to repeat once more. This is inside the nose, despite what some of you might think. And that's just a typical view of the upper septum, medial septum, the septum and the inferior medial turbinates inside the nose. And essentially control studies here showing that there are no anatomical or pathological changes as a result of a 7-day deprivation. We did 2 tasks in the scanner. One was an odor detection task. And we found that from pre to post, in piriform cortex, there was a marked reduction in the mean activity level, whereas in OFC, there was kind of an opposite effect, almost as if there's a kind of compensatory response in the OFC to support the fact that there's diminished input over the last 7 days into piriform. And both of these changes returned to baseline at the recovery time. We also looked at, did a different scan where they had to discriminate between minty smells or floral smells. And here we found that in oropharyngeal cortex, there was a change in the pattern correlation. So for example, if you had a minty smell and a floral smell, which had different sorts of patterns, as a result of this deprivation, the patterns became more similar. Subjects had greater difficulty telling them apart, as if there was kind of a tendency towards greater generalization, perceptual generalization. And we found that this effect across subjects was correlated, such that those who had a greater change in these OFC patterns have greater difficulty telling them apart at a behavioral level. All right, switching gears, as I said earlier, and often that olfaction is very much involved in regulating emotions and behaviors. We tested a specific hypothesis about whether we could modulate emotional memories in sleep. And the idea behind this was several-fold. First of all, there's recent work suggesting that one can deliver odors in sleep without arousing the subject. That's a good thing. And as I've mentioned, odors may have privileged access to emotion and memory centers of the brain. Some people think maybe precisely because you don't need to go through the thalamus, the information isn't gated in the way that other information is. That you can still, you still have access to the olfactory system in a sleeping state. And then finally, there have been a series of studies showing that odor cues delivered in sleep can enhance memories for episodic, material, and procedural memories. But no one's looked at this in the realm of emotional memories. So we did a classic Pavlovian aversive conditioning paradigm. And we did conditioning in parallel for two sets of stimuli. So here you see in the blue sector different phases, two phases. One of them is paired with an electric shock. Half of the trials it's not paired with shock. And that occurs in the background of a blue smell. It was like a faintly pleasant watermelon smell or something. The idea being is that, we'll say, for this target CS+, the conditioned stimulus, we'd be eliciting an association to an unconditioned stimulus, the US, being the shock. So that occurred in the background of the blue smell, and then the green smell, which I think was a more kind of medicinal smell. The same thing with different phases. And the blue is called target because it will be the target of our sleep manipulation. So you have the target phase that's been conditioned, and then a non-target phase that's been conditioned. So at this point, those two conditions are fairly equivalent. But then what we do is take them out of the scanner, have them take a nap for about 90 minutes or so. And while monitoring their sleep phase, every time they enter slow wave sleep, we present the blue odor, the target odor. And the idea is by presenting the context in the absence of the CS+, in the absence of reinforcement, we might actually induce a kind of extinction effect. So what happens is the target CS+ now comes to predict a safety signal or a no US signal, distinct from the condition in memory. And then finally, we return them to the scanner, repeat the same thing as before. We haven't modulated the non-target CS+. It should still retain its association to the US. But the question here is, what have we done with the target CS+? Have we changed the strength of its association to the US, or perhaps to the no US? So what we did first is we used skin conductance recordings, which measures sweat response and arousal response. And by the way, Barry, that Wonder Woman slide one was invented by William Martin Molston, or Moulton Marston, who developed essentially the first physiological monitoring system using blood pressure cuffs. So from there, he decided to move into a more lucrative department or something. So we used the skin conductance, and what this shows here is during slow wave sleep, comparing odor on versus odor off epochs. We got 30 seconds on, 30 seconds off. You can see that from the first to second half of slow wave sleep, there's been a significant decrement in the skin conductance response or arousal response, suggesting that over the period of sleep, the odor has become less arousing than before. And compared to a separate awake group, this was also significant. Curiously, there was almost kind of an inverse effect in the awake group. We're not sure why that is. So what we found is if we correlated, so each dot is a different subject, and each subject napped for a different amount of time. They woke up when they woke up. So that means each subject received a different duration of target odor. And what you can see is that for those subjects who receive more odor, there's a greater decrease in the skin conductance response, suggesting that the extinction effect, the sphere extinction effect, was very much dependent on duration of the odor delivery. And again, curious effects in awake, we found a kind of inverse response in those subjects. But what these data confirm is, first of all, the effects are selected for the target CS plus associated with the odor delivered in sleep. And secondly, that the effects were selected to sleep and not merely due to odor delivery, because we didn't see the same trends in the awake state. So we then conducted two kinds of fMRI analyses. The first was kind of the conventional mean fMRI effects. And you can see in hippocampus, which has been heavily implicated in memory and consolidation, including in sleep, that there's been a reduction in response to the target CS plus versus the non-target CS plus, suggesting that memory consolidation in hippocampus may be occurring more efficiently and more quickly, such that it goes offline from the pre to the post sleep period. And we also find in entorhinal cortex, which is very much affiliated with the hippocampus, again, a kind of dose-dependent effect across subjects. Those who slept longer showed a greater decrement in entorhinal cortex activity. The last thing we looked at was these pattern changes rather than the mean change. And the hypothesis here was the following. So in pre-sleep, as I said before, we induce associations between the target or non-target CS plus to the US. But that in post-sleep, we may be inducing a different kind of association for the target CS to a no US. And if this were the case, and we tested these pattern correlations, the prediction is that the correlation from pre to post sleep for the non-target CS plus should be relatively unchanged and fairly overlapping. But for the target CS plus, if we've induced a fundamentally different kind of memory that's being reactivated in the post-sleep phase, we should see a pattern shift. So the correlation should be higher in the non-target state than in the target state. These are just data from one subject showing that where the blue color represents greater extremes in difference, you can see that for the non-target CS plus, there's kind of a lot of light yellows, light blues, suggesting that the patterns are still fairly overlapping, whereas for the target CS plus, pre to post sleep, they're much more different. This effect is quantified across subjects, showing decline in correlation for target CS plus versus the non-target, and suggesting that there has been a kind of qualitative change in what CS plus is able to recruit as a consequence of the odor experienced in sleep. Okay, the last study, how am I doing on time?
Another 5 minutes.
Okay, I think I can shoot through this. Yeah, this is the last thing. So I spoke to you early on about how typical odors we naturally encounter are complex mixtures of many different components, but we tend to perceive them as a whole. Now, here's a wine I've never had, although Barry says he's giving it to me tomorrow. Chambertin, 1988. I don't know if any of you heard it. Unfortunately, I don't think he's got that one. Anyhow, you pour it into your glass. You can see there's a myriad of different odor components there, but in the end, we smell the wine. I mean, maybe if we're experts, hopefully we smell more than that, but it's the Pinot Noir grape, let's be honest. We smell the whole. I'm kidding, I love wine. Now, just so you know, this wine actually is foot stomped, so there's probably all sorts of curious components in the wine. Yeah. So we can use this gas chromatography technique I introduced with our carpenter ants. And we have one of these machines in the lab. So the idea is you take your compound, you inject it into the oven of this gas chromatography system, and it basically reads out all the different odors, and it can give you unique molecular signatures. So the question we ask is the reward value of a complex food odor embodied in its components, and if so, where and how in the brain? In other words, is the human brain able to read out information about salience from specific components? So we use olfactory sensory specific satiety, and here comes the ghost of Aristotle, right? We talked about at the very beginning about how it's very contextualized and contingent on bodily states. And so we can take advantage of this manipulation, as I'll explain in a second, to modulate reward value. So we ran peanut butter. I have done one of the only studies on peanut butter and jelly liking in England, because we use this same kind of stimulus with Ray Dolan's lab. And it was very difficult recruiting subjects who liked it. I'd say about 50% of UK citizens don't care for peanut butter. Anyhow, we identified 14 unique components and proceeded as follows. There's a pre-satiety task where we presented the full peanut butter smell or any of the 14 components or a control odor, which is banana. And on each trial, they sniffed, and then they either made a pleasantness or intensity rating so we could track over time those changes. We then, gave them a lunch of peanut butter and crackers until they were sated to that. That being is that by sating to the food, you would sate them to the corresponding odor of that food. We then returned them to the scanner for the same thing as at pre, so we could do a comparison. Behaviorally, not surprisingly, although always good to show, we found that pleasantness ratings for the peanut butter smell declined from pre to post, whereas the control banana odor didn't change. This is sensory specific satiety. It's specific to particular odor. As opposed to, say, if you just ate so much and you got full and you didn't want anything, you'd see decline for all compounds. Odor intensity did not change. And again, as we've shown and others have shown in the OFC, there's been a decline in the response to the peanut butter smell comparatively to the control odor. So this is a neural expression of satiety. So the question is, what about those 14 components? Here's our chromatogram with the 14 components. Kind of looks like carpenter ants. And we were able to compare, measure pleasantness changes from pre to post for each of the 14 components. There are no significant changes compared to the whole peanut butter odor. But when we looked at the orbitofrontal cortex, 3 of these components did show a significant satiety effect. And I would point out that although not significant, you can see that a decrease here, a decrease, not a decrease here. Now forget that. But if we do a correlation between change in pleasantness ratings and orbitofrontal cortex activity, 40/14 across all subjects, we do find a significant relationship, suggesting that in some ways it makes sense that if OFC is sensitive to reward value, if you take a given compound which has been reduced in value, the OFC will be tracking that. And we essentially see that sort of thing. But it shows that the orbitofrontal cortex may have access to component-based information about the value of these smells. Now, one important mediator of value in context of OFC is the amygdala. And so we also have tested connectivity between these two structures using something called PPI, or psychophysiological interactions. And what we found to the whole peanut butter odor, reduced connectivity between OFC and amygdala for the whole peanut butter smell, but not for the control. And when we looked at the connectivity for the components, so if increase in connectivity is to the right, we found that for those subjects now, for those components that showed a greater change in connectivity between OFC and amygdala, there is greater change in the mean OFC activity itself. And we also found that it's correlated with their behavioral ratings. So these are early days. This is still a work in progress, but this provides some very interesting information suggesting that the olfactory system may have access to certain components I didn't show you, but piriform cortex seems to encode the entirety of the reward value, and it has no readout of the component level changes. So we may have come across a kind of dual pathway by which the olfactory system can utilize both the configural unified information as well as elemental parts. So just to conclude, some general take-home messages. As I pointed out, for much of the animal kingdom, the sense of smell is critical for survival, and the olfactory system has evolved to detect odor stimuli in their natural form to optimize this process. The use of olfactory fMRI offers a powerful non-invasive approach to characterize limbic functional integrity of the human brain in both wake and sleep. Piriform cortex encodes odor qualities and categories and qualities in the form of distributed ensemble representations. And finally, the orbitofrontal cortex is critical for mediating olfactory perceptual plasticity and can encode salient elements of complex food odors. I'd like to thank everyone in my lab. Many of the people listed here contribute to the work I've shown. Numerous collaborators. Thanks very much to the Institute of Philosophy here at University of London. I think this Rethinking the Senses project is very exciting. Thank you to Barry and Natasha and Ophelia. This is a book I edited a couple years ago, which is more than just olfaction, but it's different author contributions, and there's a lot of kinds of topics I've discussed here about there's a whole chapter on the philosophy, the history of sensation and reward, and there's some ecology and evolution in there. Anyhow, I welcome questions. Thank you very much.
Thank you. I think before we open to questions, we'll have a commentary by John Silas, and then we'll have a question session right after that. So, John.
Where should I sit? I want to crack this up.
Right.
Someone.
Great. Well, thanks very much, Jay. That's really fascinating. I find your work really interesting. I think, first of all I really want to say is echo a little bit of what Barry said to begin with, and that's demonstrated by the work you presented here. You're in a unique position to be presenting at a workshop like this, and it's because you have a fundamental understanding. Your work has investigated a fundamental understanding of the biological mechanisms that are involved in the olfactory process, but you go beyond that and you have a kind of I think it's a cognitivist, philosophically cognitivist perspective in which you inform or look to understand better how a percept is involved in this. I mean, I think that's a fundamentally psychological and cognitivist perspective, but that's because of my background and my interest. And so it's really interesting to see a functional map, I guess, of how odor is categorized, discriminated, and identified in the human brain. Your work is really, I think, pulling that, teasing those differences apart and the different anatomical and functional regions in the brain that do that. I think really my position here is just to open up the floor for some questions and some observations and stuff that I found particularly interesting when reading your work and through your talk is this focus on binding the percept, the constructing the olfactory percept in the brain, the bound whole percept, the identified odor object, I think is how you put it at the time. That seems to me uniquely different to how we think of other sensory processes in the brain. And it seems much your work is pointing to the idea of an olfactory object forming very early on in the perceptual process, in the anatomical and functional process. And I find that fascinating, and maybe says something about a lack of the thalamus involved in olfaction, or maybe says something about the fundamental sensory receptors involved in olfaction. I'm not sure. I want to be interested to hear you speak to that as well.
I'm not sure either.
Right,
No.
okay. Great, thanks. The other thing to hear you speak about is, so I spoke to you a little bit just before we got here, is about this Noam Sobel's view and how this is, I think, quite different to Noam Sobel's view that olfaction is primarily for, or what it identifies primarily as a valence, a unidimensional kind of valence. But I think the work you're doing somewhat counter that. It's demonstrating that olfaction is more for behaviour, right? It has a particular function, and identification is perhaps the primary percept that's being formed, and not hedonic valence. I think Dick Doty would really like that, actually, the idea that olfaction is for identification as well. But there are elements in which I think you'd probably agree with Sobel, and that's this multi-sensory, this multi-modality binding that happens. This kind of, the sniff is definitely part of the percept, I think. Right? The sniff is involved in forming the olfactory object, and again, that's something to do with cross-modal binding, not just within the olfactory centre, but cross-modal binding. I think it's really interesting. So yes, perhaps the best thing for me to do is to open the floor, but maybe you'd like to talk first a bit about binding, and where you think that happens in the kind of stream.
Yeah, you brought up a lot of, should I leave this on this way? Yes. You brought up a lot of interesting concepts here. I can take any one of them really.
Right.
So in the field, there, as you mentioned with Sobel, there has been data presented suggesting that really the fundamental property of smell is its valence. And he's gone so far as to say that an odor is its valence. But I think that something like the sensory specific satiety paradigm that I showed kind of shows that that would be an incomplete account. Whatever you're doing with any stimulus, it needs to be rooted to its identity. Otherwise, you just get a haphazard spread of a physiological effect or a state effect, which is could be very damaging to the organism. I think if you think about what is the function of a given sensory system, I mean, it does seem to be fairly consistent that the approach of void distinction is very important with odor. I mean, so olfaction, we'll hear about completely different facet of olfaction next, I think, but olfaction is a distance sense. It helps you detect things from afar. And that's important because you can adjust your behavior before you end up encountering a creature you don't want to encounter or beating someone else to a food source. So that as a distance sense, it's important. But you need to have that rooted in its identification, in its object quality.
So, sorry, I don't want to take over the little, but I think just ask quickly. So is there a way to dissociate that? Is there a way to pull apart? So I know actually it's been done in frontal asymmetry, DEG, social cognition type stuff, but is there a way to do it with olfaction to pull apart valence versus this approach withdrawal in the olfactory system?
Well, I mean, people like Ken Barrage talk about wanting and liking in animal models. One can debate about whether those behavioral assays really nail down those distinctions. But, I mean, it partly depends how you operationalize those concepts in the first place, because, you know, when someone becomes sated to the peanut butter smell, I guess, you know, actions speak louder than words, and if they don't want to eat anymore, they must not like it anymore. But you could say, well, if you put that aside for a minute, do you still like the peanut butter? Do you regard this as a smell that you like in the universe? And they may say yes, right? So, but it just becomes a little clumsy. What I would say is, I think we do need better ways of approaching these questions. They're difficult. You know, animal models have many advantages, but at the end of the day, you know, that's all very indirect ways of assessing these questions.
Okay, so let's come up front and then we'll take questions from the floor. Matt. Yeah. So I had a question about the representations of categories that you described. Are those representations created by patterns in the stimuli, or are they driven by, as it were, top-down processes of us categorizing stimuli in various ways?
That's a very good question that we don't know much about. The fact of the matter is, if you look at the projection patterns from the receptor neurons to the bulb, it's highly convergent. But then when you look from the bulb to piriform cortex, they're highly divergent. So one given cluster point in the bulb, which is encoding information about odor in X, will project widely to piriform cortex in a fairly haphazard, disorganized way. I mean, it's just disorganized that there's no topography, there's no clear spatial relationship there. It may be preserved, right, within that subject, but it's very distributed, much in the way that you see information coded in the hippocampus, also in a kind of pattern distributed way. It's something called content addressable memory system, where so you know, remember, we don't tend to encode. Well, let me say that an odor stimulus doesn't really contain spatial information within it. You may find left-right nostril differences, but that's a much cruder level. We don't map the olfactory world like we map the visual or the auditory world. So in some ways it doesn't make sense to have a kind of rigid spatial-based topography. But you do need to have a preservation of the information anyhow. So these patterns are probably driven as much by the bottom up as the top down in some ways. There's a very rich kind of associative feedback structure or circuitry in the piriform cortex as well as in the hippocampus, which can help create these patterns. And one benefit of pattern-based codes is they're very resilient. If you present part of a material, in the natural world, you don't always encounter the same archetype stimulus every time. If you encounter something that's kind of partial reconstitution of the whole, those patterns can kind of reassemble the whole in a kind of computational way. Those patterns can perhaps more easily generate other exemplars of the same, other exemplars of that stimulus, and may be well suited towards reactivating the stimulus in terms of memory. So, obviously the top-down will also play a strong role. I mean, you asked a very good question, and it would be really, we've actually, so there's the bottom-up and there's the top-down, and we've actually begun doing a pharmacological study to try and block the top-down using a drug called baclofen, which is a GABA-B agonist. In some ways, in that odor deprivation study, we were able to block the bottom up. And so now we're trying to take, but it's very difficult. I mean, in some ways, there you really need animal models, so you can do more selective lesions or pharmacological blockades. But that's my view of patterns.
I had a couple of questions on your sleep extinction study. So first, when during the conditioning, the odor was consciously perceived, or
During conditioning itself,
yeah.
It was in the background. It was a super threshold stimulus, and they had a nose piece, so they were aware of the odor, but that wasn't their focus. They were focusing on perceiving the face stimuli, and I suppose worrying that they were going to be getting shocked.
So if that's the case, then the extinction when you just give the blue smell again without.
So
it's a non-conscious one. So you have the extinction phase is with
during sleep.
During sleep is with non-conscious.
You could say that, yes.
Okay. That's
Yeah.
nice. And the second thing, you call it extinction, and you said two things which are a bit sort of different. So I was wondering which one you exactly think. One is that because you reassociate the smell during sleep with safety, that it sort of counterbalances the first sort of conditioning. And the other interpretation is just there is no bad stimulus to bind it with anymore. So it's just like an absence that will do the extinction, or it's a new learning with safety. So which one is it?
So you sound like perhaps you know a lot about this area, but there's kind of it's not really controversy much anymore. The question is, what is happening during extinction? And there are two camps. For many years, people thought that it was essentially an erasure of the original fear. So once you had it, now you don't. But there were more and more studies, mostly in animal models and more recently human, showing that in the right context, you can fairly spontaneously bring back the fear memory. Okay, so they've been extinguished, but in the right circumstances, you can bring back the fear memory, suggesting that it was never erased in the first place. At the same time, there are studies showing that there is the formation of a unique memory trace, the safety trace, that is encoded qualitatively differently, perhaps with different sets of amygdala neurons than the fear memory. And so in some ways our hypothesis was testing that model, if there was simply kind of a relaxation in the strength of that fear memory, we might have expected to see the same pattern, right? You know, even if it was even if those neurons or those voxels were reduced in their net activity, the patterns would be the same because it's the same populations being utilized, as opposed to this new safe memory, which would have been a qualitatively distinct trace, and that's what we found.
Okay. Colin?
I'm interested in the fact that it's usually, as you said, difficult to distinguish the individual components in complex odours, and that they're experienced more as being representative of the thing that's produced them, so coffee being the obvious example. First, does that apply to unfamiliar odour mixtures? Is it also impossible to distinguish the individual components of those, and do they achieve some kind of status as an object of perception? I mean, the interpretation that you could put on this is that, yes, there's very early development in the olfactory system of an identified experience, but another is the analogy with colour vision, where the inability to distinguish the individual components of wavelength mixtures, they simply can't be distinguished at all, and identical experiences can be produced by very different mixtures if they stimulate the receptors in the same ratio, proportion. Couldn't that be true also of olfaction? And I know that there's a long history of looking for primaries in olfaction, the possibility that the very wide range of olfactory experiences that we get is actually made up from ratios of activity amongst some kind of primary elements. So it's a long question. Can you tell us about that?
So one way, as we've been thinking about that data set in the lab, I think one interesting parallel to the visual system wouldn't be color vision so much as, let's say, face perception. So, for example, right, we perceive the whole face, and there's many neurological syndromes in which we cannot perceive the faces. But there's behavioral and imaging evidence suggesting that there are elements of that face that carry a lot of information. Even the eye tracking data showed that we fixate much more on the eyes. And in terms of emotion, let's say, and we're dealing with value, right, more than the odor per se. A lot of the value information in face is read out from the eyes. And so I guess that's how we're kind of conceptualizing what we may be seeing here. I mean, one interesting question is why those three components. A couple of them are pyrazines, which have a very nutty smell, but they're not very peanut buttery smelling. Another one has a more kind of roasted note. But I agree with you. In terms of the familiar, unfamiliar, I think that, so there's two ways to consider it. Either, let's say those three components are innately rewarding for some reason, or it's been learned through experience. And I think it's the latter. And even there, so what would happen if we took those three components and put them in, let's say put them in an otherwise unfamiliar smell concoction? Would that still have some kind of, would that have a more inherent reward value? And that could be an experiment we should probably try.
Pyrazines are often quite volatile. They come off early, and they're winemaking, you try to guard the primary aromas in, say, Sauvignon because it's pyrazines. So maybe a measure of freshness. So finding something that's fresh and then disliking it later. This is certainly the most volatile. If you've got the aldehydes, you've got the pyrazines, you're going to get the pyrazines spiking much higher, I would have thought, consciously. So you could do it. But Colin's question, you've got coffee and you've also got the smell of the sea, which people recognize, amazingly complex in terms of the number of molecules in there. But we can have very complex wines that we've never experienced before, and people still have difficulty dissecting and finding what the components are. So even if it's unfamiliar, it can strike you as... What's interesting is that you can recognize it as complex and you can't recognize what the parts are.
But, right, and none of our subjects were identifying components, but the brain is, apparently. And, you know, maybe because the olfactory system kind of divides so early, and it's a very, I showed one slide, I didn't dwell on it, but from the olfactory bulb, there's probably parallel information projecting to about 5 or 6 structures. The chief one is to the piriform cortex for extracting the object information. But, you know, within 2 synapses, there appears to be, and if you look at recent animal tracing studies using optogenetics, there are fairly tight clusters of projections from the bulb to discrete areas of the amygdala. Now, any one glomerulus in the bulb is effectively encoding just one odorant information because of this convergent projection pattern. So the amygdala theoretically does have access to that component information. I don't know in the visual system, if we go back to color, whether there is that availability fairly early in the system, or whether it's integrated so quickly that it's just not accessible. But there's something about the olfactory system which could make that possible. And you know, this work didn't occur in a vacuum. There's work in both mice and moths showing that there are particular components of either flower nectar scents or, say, mouse urine pheromone that are particular triggers for the behaviors. If we can only ask them if they're physically, perceptually recognizing those components, that would really...
But even though the subjects aren't recognizing components consciously and behaviorally, subjects make judgments, not always accurate, but about whether it's a complex odor or not. So something is going on below the level of consciousness that puts a flag up that says complex or not. But we can have single molecules that people think are complex. You can have 2 molecules people think are complex, you add a third, they think it's simple, so we know you can go wrong. That there's something about the components being assessed and that it's categorized as together or not together. So is that environmental? Is it
Yeah, I don't, you know, Sobel, who was mentioned before, has this idea that beyond about a mixture of 15 odors, it all starts smelling the same. Like maybe you're swamping the receptors to such an extent that you get a kind of noise signal, which they call white. I know, they would say, well, you have 613 components, but really only 2 of those are abundant enough, or 2 of those carry most of the message. But
who knows? Okay, I'm going to take 2 quick questions, then we must stop. One from Keith and one from Adrian.
I want to ask about the actual receptors. You showed us these wonderful pictures of the mats of cilia, which I think feed the olfactory area, so the kind of receptors. How much specialization is there in the different, in the eye, for example, we have, you know, basically 2 different kinds of cells and 3 different kinds of colours respond to it. Are each of these picking up on different chemical properties, different kinds of substances? Are they highly specialized? Or what do we know about that?
Yeah, that's a really good question. So first of all, let me caution you by saying that we don't know anything about the human brain, especially with regard to the question you just asked. So in rats and mice, which express 1,000 functional olfactory receptors, olfactory receptor family is probably the largest distributed gene family in the genome. So there's 1,000 unique receptors, but for any given receptor neuron, like I showed you in the freeze fracture, for any given neuron, it only expresses one receptor gene. So all those cilia from that one olfactory receptor are expressing the same gene. So they're all tuned to the same odorant. And when I say odorant, that's an odor molecule of physical stimulus. Now, and so if there's, I don't know, many millions of those olfactory receptor neurons, and there's 1,000 unique receptors, then that means what? There's 1,000 or 10,000 or 100,000 neurons expressing any one of those receptors. And those are distributed more harmoniously through the receptor epithelium. There may be some loose clustering in some ways. There's kind of 3 general classes of olfactory receptor neurons, but beyond that, they're very homogeneously distributed. And what's interesting, and when I talk about the convergence from the receptor level to the bowl, so you have those 100,000 receptors all encoding the eugenol receptor. All of those receptors project to the same 1 or 2 glomeruli in the bowl. That's massive convergence. And part of that may reflect the fact that the olfactory receptors are expressed on the cilia, but also all the way through the cell body and out through the axons into the bowl. And they serve as a tracking mechanism for wiring. Right? If you express together, you converge together, sort of thing. And it's interesting why there is that step, because then you disseminate the information again in a very scattershot way. But whether these principles apply to humans is unclear. There's basically 1 to 2 ratio of receptor types to glomeruli, because any one projects to 2 glomeruli. If you do glomerular counts in the human bowl, that ratio changes from 1 to 2 to about 1 to 10. So there may be a very fundamental difference in how we are organizing the olfactory space compared to rodents.
Thank you for your excellent talk. I was very interested in your research about the smell memory during sleep, and I want to know your opinion. Could this be used as a kind of sleep communication? So, for example, learning during sleep based on awake learning, enhancing that, or in affecting dreams, or some kind of sleep communication. Would that be possible?
Right. I would say that the answer to that is yes, but it depends what kind of information you want them to learn. And the Sobel lab is also doing some work on smell and sleep, and they've shown that if you pair a tone and a smell in sleep, and if you use the size of their sniff or their respiration as an index of learning, that in the post-sleep period you find these respiratory differences when you present the tones. So that's a form of learning. Now, if that's where you want them to learn, I guess you can do that. The idea of putting a textbook under your pillow doesn't work. And I mean, it's been done. So I didn't mention it. If you have subjects learn a bunch of picture-object location associations. There's a grid, you learn one object's there, another's there, and so on. And you do that so some of those objects are paired with a smell and some not. If you then present that smell during sleep, afterwards they have better memory for the locations of the objects carried. So it's very much a dynamic field right now, and not just odors. Tones also seem to have access to neural circuits in sleep. So there's a lot, it's really kind of a burgeoning field. I'm hosting a symposium at the annual Smell and Taste meeting, which is held in Florida every year on sleep. We're going to have a Drosophila person, a rodent person, a human person, if you find yourself in that area. It's a very exciting area right now. We're looking at a few more of that.
What about what about the dream? Could we program the dream, make happy dream or fearful dream based on this smell?
But people usually report they don't smell dreams.
But if the smell affects the memory, maybe it could affect the dream emotion.
I think part of that is because a lot of people are fairly ignorant about their olfactory experience. I have smell dreams.
So does hand go up from Sarah?
The Professor Thomas Hummel said, I think he said 5% of people dream in smell.
I mean, for me it's not frequent, but like I've had smell and taste.
I will present some data on that later this afternoon.
Oh, wow.
Okay, let's not preempt you then. That's going to come on. But so we're going to move straight on, just a sort of 2-minute comfort stretch while we set up the next talk, and I will hand over to my colleague Colin Blakemore to chair. But before that, let's thank John and Jay very much for sticking with us.